Hello everyone, this is Rosario, second period honors biology, and this is my DNA replication model. So the process of DNA replication happens in the nucleus in eukaryotes. When talking about DNA replication, we have to talk about the structure of DNA. DNA is a double helix or twisted ladder. DNA is also double-stranded. The backbone of DNA consists of a 5-carbon deoxyribose sugar and a phosphate group. The rungs of the DNA molecule are made up of 4 nitrogenous bases: thymine, adenine, cytosine, and guanine. These nitrogenous bases pair up together to create the rungs of the twisted ladder. However, only some bases pair with each other. Through Chargaff's rules, we know that thymine only pairs with adenine, and adenine only pairs up with thymine, and cytosine only pairs up with guanine, and guanine only pairs up with cytosine. This is what is known as complementary base pairs. The nitrogenous bases, along with the deoxyribose sugar and phosphate backbone, come together to create what is known as a nucleotide, which makes up the whole nucleic acid of DNA. Adding on, the 2 strands of DNA run anti-parallel from each other. While one strand runs 5' to 3', the other strand runs 3' to 5'. There are some important enzymes in the process of DNA replication. There is helicase, primase, DNA polymerase, exonuclease, and DNA ligase. So the first step of DNA replication is for helicase to unzip the double-stranded DNA molecule. Then the enzyme primase will place down an RNA primer at the start of the DNA strand. Then, DNA polymerase will start adding complementary base pairs starting from the primase from the 5' end to the 3' end. This is known as the leading strand, as the DNA polymerase is able to continually add bases to the template strand. However, for the other strand that goes from 3' to 5', it has to do it in little tiny sections because DNA polymerase can only add bases from the 5' to 3' direction. It starts with primase adding on an RNA primer. Then DNA polymerase will add on bases starting from the primer going in the 5' to 3' direction. Then the enzyme primase will add another RNA primer further down the line of the 3' to 5' strand. And DNA polymerase, starting from the primer, will add on more complementary base pairs going from the 5' to 3' end. These small segments of the DNA strand are called Okazaki fragments. Then, the enzyme exonuclease will remove all of the RNA primers. Then, DNA polymerase will fill in the empty gaps with complementary base pairs. Finally, the enzyme DNA ligase will act like a glue and glue together the empty gaps. Final product of DNA replication is 2 exact same copies of DNA. The end.